Thank you for joining us in today's webinar. Today, before I proceed to introduce our speaker, Olivia, I want to bring a couple of announcements. Remind you that we have awards for our champions. These are for $2,000, and it's a year commitment for active members that do take on leadership roles such as the chair or co-chair of a working group, lead manuscripts, or advance some key deliverables for the working groups. You are supposed to have active participation and quarterly check-ins with the project coordinator, currently me. And at the end of your journey, you write a blog post for this. There's a QR code for more information, and there is also, we have the education awards. This is to implement or develop on the educational curriculum that the education working group brought together, and these are for $600 for particular tasks. It can be to implement the curriculum into existing courses, add new modules, or revise and translate or translate existing ones. From the workshop, we have a volunteer, Elsa Quesada from Mexico, who's willing to help translate these, but she unfortunately is not affiliated with a U.S. institution. So if you want to pair up with her, that she's very happy to help us with some of these. And here we are. So it is my pleasure to introduce you to Olivia Haley. Olivia is an ORISE postdoc. Research fellow at MaizeGDB. She did her master's in plant science at McGill University and her PhD in horticulture and natural resource sciences at K-State. Her research focus, also those of you that were at the workshop might have seen her picture somewhere there, because she was highlighted in her program. Her research focus is in machine learning and computational biology for protein functional annotation. She develops AI-ready genomics workflows and scalable data infrastructure. She also uses protein language models and structure-based tools to reduce manual genome curation. And she's very passionate about accelerating discovery through AI in plant science. A fun fact is that she is originally from the Palmetto State, which I just learned about, South Carolina, and she enjoys growing southern crops in the Midwest, which is a very different climate. And she's going to be telling us about how she's working on the basis to transform maize data into AI-driven discovery. And with that, I'm going to stop sharing. It's all yours, Olivia.
All right. Thank you so much, Marcella. Let me share my screen. There we go. Okay, so can everybody see my screen?
Yes.
Awesome. Perfect. Well, thank you so much, Marcella, for that introduction and for inviting me here. I'm really honored to be here. Recently, like Marcella said, my name is Olivia Haley, and I'm an ORISE postdoctoral research fellow working with MaizeGDB. And so recently, I've been assisting in MaizeGDB's AI Ready initiative, which means that not only is the data that we generate adhering to FAIR principles, but that it can also be readily integrated into artificial intelligence and machine learning pipelines. So for today's talk, I'll just be presenting a short primer on what is MaizeGDB, the criteria that guided our AI readiness initiative, the new data sets that we generated, and then how we're using those data sets to develop new tools or improve our existing tools that improve our understanding of functional analyses and biocuration in maize. So the Maize Genetics and Genomics Database, or MaizeGDB, is a community-oriented USDA-funded informatics service that is focused on the crop plant and model species Zea mays. So we provide highly curated data, valuable informatics services, and actively build tools to visualize and analyze maize omics data. So this screen just shows a snapshot of the data that we're actively managing and building tools for. And these tools are all managed, these tools and data are all managed by a team of 4 permanent USDA scientists, or excuse me, USDA employees, led by Carson Andorf and 3 postdocs. There we go. Sorry, got stuck. And like many agricultural databases, one of the challenges that we've been facing is just the explosion of AI in the bioinformatics space. So, for example, thanks to AlphaFold 2, whose creators won the Nobel Prize in 2024, we now have an immense repertoire of predicted protein structures. And there's also been, as I'm sure you've noticed, just a plethora of large language models that have shown promise in assisting with various stages of analyses like biocuration. And there are even these more specialized implementations of large language models called protein language models and DNA language models that can help us predict if a protein is functional or not, help us identify genomic elements, and then score functional variants. And many of these advancements have come within the last 5 to 6 years, which just demonstrates like the breakneck speed at which AI is developing in genomics. And so, with this rapid pace of development, our goal is to ensure that MaizeGDB is strategically positioned to be at the forefront of these developments of AI and plant genomics, or in other words, to ensure that we are AI ready. And so, first, it's important to distinguish how we define AI ready and how AI ready adds to the already widely accepted FAIR principles. So, FAIR principles refer primarily to the ability of the user to easily find, interpret, and reuse data, whereas AI Ready adds another layer onto this and emphasizes having data that or structured data that is enables users to develop models that learn or generalize biological significance. And so a data set can be fair, but not AI ready, which means that it's well documented or really noisy and not well structured. Or data can be AI ready, but not fair. So you have a set of gold standard, well structured data, but its provenance is not well documented or it's not publicly accessible. And so, using these definitions, we outlined our working vision of AI readiness using 3 primary criteria. And so our first criteria tackles file formatting. So as many of you know, omics data can be highly inconsistent with so many different data types like FASTA, FASTQ, BAM, BigWig. And then also so many different data types stored within a single file, and not necessarily, for example, like the column values, not necessarily being the same from file to file. So, our first criteria was to create structured, standardized, and reproducible data, which involves making sure that the data is in a consistent format, that it uses consistent descriptive column names and controlled vocabularies that limit ambiguity, and then also that the data supports version schemas for reproducibility. And so this represents a shift from FAIR principles where the priority is mainly for the data to be in an accessible, kind of more human readable format, to on top of this, the data also being in a format that prioritizes machine readability and easy integration into machine learning workflows. And so the second criteria tackles a major challenge in biological data, which is noise. So biological data is inherently noisy. And there are many different sources of variation. So, maybe you did your greenhouse experiment at a different time of the year, or maybe you used a different brand to perform your assay. So, because of the power of techniques like deep learning to model and sometimes reproduce a variation, this means that you can actually accidentally start modeling your data's noise rather than modeling your true biological variation. So, our second criteria just involves minimizing this bias, particularly in pre-computed data sets, by clearly marking or separating sources of outliers. So, for example, non-coding RNA, making sure that your data sets have informative features that machine learning engineers can use to participate or partition data into train and test data, and then delineating between experimental data and computationally inferred labels. And so this shifts the focus or yeah, shifts the focus from not only documenting where the data came from, who generated the data with what version of software, but also preemptively controlling for different sources of noise that can be seen by machine learning models. And the last criteria addresses making the pipelines and the data available for reuse. And so you may be thinking that, okay, FAIR principles already cover this, but I'm sure many of you have tried to reproduce analyses from a GitHub repository only to find that the code requires so much compute that it is practically inaccessible. So our third criteria addresses this point, and starting with providing well-documented resources for reproducing data sets and features through platforms like GitHub. And then we also reduce the computational burden of our users by providing pre-computed data sets using pipelines that can only be generated using GPUs. And we also created a platform called Maize Feature Store that allows for large scale feature extraction. So users can build custom data sets. And so these criteria not only encourage data sharing, reuse, and attribution, but on top of this, support the adoption and use of machine learning within the community. So, taken all together, these are the criteria and the sub criteria that have guided our initiative. And now now that I've defined this sort of what and the why, I'd like to shift more towards the how. So, I'll show you three main ways that we're fulfilling each of these criteria that are on our list. And so, solidifying our foundation of our AI-ready infrastructure didn't always equate to creating a new data set. And so, the first was by establishing a consistent schema and file standards. So, and this is particularly was particularly interesting in our case because MaizeGDB hosts data from before it was officially established in 2003. So that meant that some of our data not only predates our database, but also predates contemporary file formats and standards. So we sought to make sure that these file formats were consistent and that they contained consistent data structures and that they were all well documented. So in this table, there are just a few examples of the file formats that we're committed to using consistently for each of the data types. And we also already have much of our analyses documented through our GitHub, which supports versioning and public sharing of workflows within our community. So by making these repositories public, we can share or we can contribute to building community resources and making sure that our data stays reproducible. And so this is just an example of a subset of our public code repositories that can be found on our GitHub. And so one of the more time intensive tasks that we took on was actually curating and creating new data sets that are filled with standardized gold standard data. So this table provides a summary of our data sets that we already had on hand. So like our protein embeddings and our predicted protein structures, and then also the data that, or the data sets that we either constructed or reconstructed for our AI readiness initiative. So our variant effect scores, our core 60 vector, our extended 17K vector, and curated literature data set. And I'll be diving deeper into this topic because having access to these data sets or these gold standard data sets are at the core of machine learning and artificial intelligence. So in 2000, so to this point in 2024, so this figure here shows data from a 2024 survey from Anaconda, in which they're asked over 1,000 data scientists how they spend their time with data. And their respondents answered that on average, 67% of their time is spent preparing, cleaning, visualizing, and presenting their data. And so that's 67% of the effort even spent before even selecting or testing different models, which in my opinion is the fun part. So in particularly, if we look at the distribution of how much they're spending on just preparing and cleaning the data, we find that almost 40% of their time is spent within data wrangling. So understanding your data is definitely fundamental to making meaningful insights. But fields like computer vision and computational chemistry have been able to accelerate their AI efforts using standardized data sets. And these pre-cleaned, predefined data sets save time on data preparation and cleaning, accelerate hypothesis testing, and set a universal standard for evaluating machine learning and AI models. And so, a great way to demonstrate this point through efforts that we're doing at MaizeGDB is through our precomputed protein and DNA variant scores. So these zero-shot scores are computed by protein and DNA language models, which are trained to take a sequence and predict the identity of missing or masked base pairs or amino acids within the sequence. And so this allows them to learn sequence conservation patterns. And these learned patterns are then transmitted as probabilities of different substitutions appearing within a natural population. And so these probabilities are used to calculate log likelihood scores or zero shot scores that predict the likelihood that a substitution is or has a negative impact on the function of the eventual protein that's being produced. And so, and more negative values are suggestive of more deleterious mutations. So these models produce powerful metrics to predict functional consequences of allelic variants. And we've also integrated them into our various tools, such as our SNPversity tool, which surveys 250 million single nucleotide polymorphism sites across 3,000 Maize accessions and shows the predicted functional consequence of each SNP. And we also have developed PanEffect, which is a tool that visualizes the protein variant scores corresponding to every possible substitution in the B73 proteome. And so now I'd like to give you an example of how the SNPversity tool, which integrates these DNA and protein variant scores, can be used to accelerate plant breeding. So maize sugary 1 encodes a starch debranching enzyme that is defective in different varieties of sweet corn. So this mutation or mutations in this protein result in a kernel that has a higher sugar content and a wrinkly or wrinkled phenotype. And so that's what these photos here are showing, the various different phenotypes of mutants with the images available at MaizeGDB. And the table to the right is just a summary of these SNPversity results for this gene, which includes the reference base pair, the allele variant that's seen within the natural populations of maize, the resulting point mutation within the protein, uh, the DNA variant effect scores calculated by plant and then the protein variant scores, which are calculated by ESN variant. Um, and then this table also shows the variety in which or the mutation that is seen. And what's kind of interesting and really cool is that these scores reflect experimental data that we have on these point mutation variants. So, for example, these variants that are highlighted in red were naturally seen within the population, or within the population of sugary 1 mutants. However, the literature found that they don't actually result in a substantial change in the protein's function. And what's cool is that our DNA, the DNA DNA variant effect scores and the protein variant effect scores also reflect this, and that they're higher than the effects within the population that are more consequential, which are these other mutations. And the variant effect scores integrations on our JBrowse tracks also show a close alignment between the peaks and genomic elements, like the coding regions, uh, the coding regions, and then also the transposable elements, which indicate that the DNA language model that was used to calculate these scores, so PlantCAD, can detect a wide range of variation within the genome. There we go. And so beyond our variant effect scores, we also sought to contribute downloadable gold standard datasets to our AI-ready infrastructure. And this initiative actually began a few years ago when we released Maize Feature Store, which was a collection of approximately 17,000 pre-computed features for the gene models in our reference genome B73. And this includes features like intron exon length, amino acid composition, and then more complex features like WGCNA principal components. And since then, these features have been used to build machine learning models that identify phenotype associated genes and predict pollen fitness. And we also developed a feature vector of just the top 60 features, which includes data spanning across the gene structures, tissue mRNA and expression features, and protein structure features, and protein biochemistry features. And so while some of this data is available only for the reference genome, B73, most of these features are available for all of our highly curated nested association mapping or NAM lines. And so these features were also decided on based on their availability within the data corpus that we already had, and also their use within the machine learning literature. So, for example, predicting tissue-specific mRNA and protein expression is a major area of research where machine learning is used. So, we've included various features that can be used to complement this research. And so these data sets are useful for multiple reasons. Since they're standardized, they can be used to develop machine learning models. However, they can also be used for non-machine learning purposes like performing descriptive statistics on gene sets, as well as functional enrichment. So even if you're not a machine learning practitioner, these data sets can still help you discover insights and test hypotheses. And so one of the ways that we've demonstrated how these data sets can be useful is by using them to develop a platform for functional enrichment, which is called FETA, or functional enrichment term analysis. So, the functional enrichment analysis is among one of the most widely used methods in bioinformatics. However, we found that for Maize, most of the currently existing platforms and tools only allow you to do functional enrichment within our reference genome B73. And they were really restricted to only using two types of ontologies, such as Go term enrichments or pathway enrichments. So leveraging our core 60 vector and other data at MaizeGDB, we designed Feta to allow users to perform multiple simultaneous enrichments for reference and non-reference populations using ontologies that focus on biological function. So for example, Go terms, biological pathways, metabolic function, protein domains, but also on covering trends in single cell and spatial distribution. And the overrepresentation, this overrepresentation analysis can also uncover other trends within the data set. Sorry. So here, for example, is where you can do B73 and non-B73 enrichment. And then, yeah, so we also have available overrepresentation analysis in other using other types of metrics such as conservation, evolutionary conservation. And we've also recently expanded its use to allow users to perform enrichments on differentially expressed genes, or DEGs, that are identified through popular transcriptomic pipelines like AgR, DESeq, Lima, etc. And so this platform allows users to upload their pre-processed data from AgR, for example, visualize their differentially expressed genes, and directly perform functional enrichment on the subsets, all in one place. And so I just wanted to provide you with a concrete example of how this platform built on our AI ready data can do something that other enrichment platforms can't. So the data set that I use for this analysis comes from a paper of Shurak et al. 2021, who compared the transcriptomic response of a corn smut resistant line, CML 322, to a susceptible line, or B73. And so what we did was that we took their 431 upregulated genes from CML 322 and ran them through Feta. And so the original authors, so in the original paper, they noted an enrichment of oxidation reduction related Go terms, and hypothesized that these either reflected ROS, or reactive oxygen species detoxification, or some sort of relay signaling. And so using Feta, we were able to go even further and run Go term, enzyme commission, PFAM domain, and subcellular localization enrichments, all at one time. And so the PFAM results, which you're seeing as a network here, connectivity or CNET plot, pointed to an overrepresentation of membrane-bound proteins that had NB ARC domains and protein kinase domains, which are the hallmark architecture for a subset of disease-resistant genes called NLRs, NLR-RLKs. And so this CNET plot shows the connectivity of these overrepresented PFAM domains. And so what's kind of cool is that four of these proteins with this characteristic of architecture actually mapped to a locus for Puccinia sorgi resistance, and then two others were previously identified in the literature as candidate genes for southern rust resistance, and then one other also was hypothesized to encode for RPM1, which is a resistance gene for Pseudomonas syringae. So using our AI-ready data, we were able to build an application that takes gene lists and within seconds surface a sort of mechanistic hypothesis on what they do. And so, from an AI readiness perspective, Feta could be used to characterize genes that maybe an AI model consistently doesn't generate good predictions for. And so, this could be helpful in identifying whether there are statistically significant shared functional pathway or domain level patterns in the mispredicted gene models. And so this kind of systematic trend analysis could also help researchers understand why certain genes are difficult for AI to handle, and potentially guide improvements to model training or annotation strategies. And so the last data set I wanted to talk about was our curated literature data set. So biocuration, as many of you know, is the process of extracting functional annotations from the literature or experimental evidence. And so curators are invaluable to genomic databases, but curation can take a significant amount of time. So there's growing interest in leveraging large language models like ChatGPT and Claude to assist curators. But to train and evaluate the efficacy of these LLMs, we have to have a gold standard set of curated literature. So because MaizeGDB has a long history of manual curation dating back to the 1920s, where this image is actually from, we created a data set that contains around 5,000 publications that have some sort of manually curated annotation. And so this includes crop traits, metabolic pathways, phenotypes, variations, or functional variants, etc. And the hope is that this data set can be used to help refine these large language models, or serve as a baseline for testing the efficacy of different machine learning based biocuration methods. And so one of our postdocs, Laura Tibbs Cortez, sorry, Laura, is collaborating with Kapil Chugale of Sorghum Base and the NLP for Biocuration Working Group to put this data set to use. So one of the problems with current AI workflows is that they aren't really domain specific enough. So they aren't usually able to connect the dots between labels like gene models to variants to crop traits, this very complex network. So she's currently exploring the using our curated literature data set as a baseline for evaluating a machine learning based biocuration pipeline called Texpresso. And so Texpresso, takes in a PDF document, it tokenizes the text, and then performs various analyses to extract the annotations. And so this protocol can save biocurators so much time on extracting annotations like gene models and traits, methodologies, and gene function. And so much of the contents of this presentation is actually in our recently published paper, where we discussed our AI readiness initiative in more detail and provided more use cases for our AI integrations at MaizeGDB. And so if you aren't able to access the paper, please feel free to reach out, and we can help you get access to that. And lastly, I just wanted to say thank you again to the MaizeGDB team. I'm just so fortunate to be a part of MaizeGDB, and really to have access to all of their knowledge and support. And so now I'll take whatever questions you might have. Thank you.
Thank you, Olivia. That was a wonderful presentation.